Alright, good afternoon, ladies and gentlemen, other welcome guests. Thank you for coming here to see as we present our 3-work research project entitled Duckweed, Dual Platform for Renewable Fuels and Wastewater Remediation. I am Jay Wanaba, this is Anne-Marie Barrett, and Nikita Dave. We will begin by discussing the ubiquitous crisis that affects the globe right now, fuel needs. So this impending disaster can be categorized into 2 major aspects, underground and above ground. The former mainly deals with natural gases and fossil fuels and their recent depletion due to exponentially rising demands for oil and energy. So, peak oil is the main extraction point of oil before its complete depletion and exhaustion. So, this has in turn caused many fears to travel above ground, such as Middle East tensions, where prolific oil reserves lie. So, alternative sources of energy are being delved into. America has attacked this problem with the introduction of ethanol-based fuel. However, the primary source for ethanol, being corn or maize, comes with many drawbacks. The first that it's a necessary food stock for us. That's its first usage. So in its conversion to ethanol, other issues lie. Many pesticides are used, resulting in toxic runoff and soil erosion. Also, to optimize its conversion, you must use crop rotation and appropriate waste management, which inevitably heighten the carbon footprint, which is very counterproductive to its initial usage. So, carbon footprint is a total emitted greenhouse gases that can affect the environment. Inoculation is placing a plant or a bacteria in an environment where it can grow. In our scenario, it will be duckweed in different forms of media or water. Media is a liquid that supports the growth of a plant or bacteria. And finally, incubation is to place an organism in a controlled environment.
So with the increased demand for crop production, as Jay was saying before, specifically corn, there's an increased usage of fertilizer, and thus other types of runoffs and lead to wastewater, and it's just not good overall. So the issue is wastewater remediation. So the EPA, the Environmental Protection Agency, has actually placed limits on wastewater effluents. And effluents are just wastewater discharges to surface waters. So while these limitations and guidelines currently exist, wastewater remediation and purification is actually very difficult, much more than you would assume. So current ways to remediate wastewater include physical means such as sedimentation, chemical means such as chlorine usage, etc. But a new source has to be developed. See, these waters will contain high levels of nitrogen, ammonia, phosphorus, nitrates. All of these things are going to lead to eutrophication. And eutrophication will result in destruction of ecosystems, water sources, and ecosystems surrounding them. See, what happens is the biological productivity will actually be disrupted from these high levels of nutrients. So in comes duckweed. So what exactly is duckweed? Duckweed is the smallest flowering plant in the world, and it is roughly 1/4 of an inch in diameter because it is circular. So it is roughly 93% water by biomass, and it actually is known for its prolific abilities and will grow in abundance in just about any still water or any sort of wastewater. So the amazing thing about duckweed is that it has a high percentage of starch, which can be made into dextrose and then thus ethanol. So another source is that duckweed actually will thrive in environments that are enriched in those sorts of nutrients such as ammonia and nitrogen, and can hopefully curb the eutrophication process.
So our experiment serves to compare the efficiency of ethanol production from corn to duckweed. This can be done in a multitude of ways. However, aside from researching the difficulties of using corn as an ethanol source, such as its unpredictable prices due to its dependence on weather conditions and its competition as a food source, we decided to conduct an experiment to determine the percentage of starch in duckweed. Now, using percent starch and other factors, we were able to effectively compare both corn and duckweed to determine which one could produce a more viable biofuel. Also, we evaluated the efficiency of using duckweed as a wastewater remediator, as it has very long roots with much surface area that are able to absorb many nutrients and toxins from wastewater. To test our hypothesis, we conducted 3 experiments. Our first experiment, the biomass growth experiment, we correlated high nutrient levels with high biomass accumulation. In our second experiment, the nutrient content examination, we analyzed ammonia levels of inoculated duckweed before and after 1 and 2 weeks into the experiment. In our third experiment, the starch assay, we determined the average percentage of dextrose in duckweed. Dextrose is a monomer of starch and a variation of glucose that is used to produce ethanol. The purpose of the biomass growth experiment was to determine which species grew the best in which media and how nutrient levels affected this growth. Setup included using 7 species of duckweed inoculated in 3 different media. Media meaning wastewater from 3 different locations: Princeton Meadows, Pine Lands, and Passion Puddle. The original masses were then later compared with the masses of duckweed, inoculated duckweed 2 weeks into the experiment. So the water samples taken from Princeton Meadows originally had 0.2 milligrams per liter of ammonia, whereas the water taken from Passion Puddle and Pine Lands had 0.1 milligrams per liter of ammonia. The effect of greater initial concentrations of ammonia is illustrated in the fact that there is much higher percentages of growth in the duckweed inoculated in the media from Princeton Meadows. Both the Passion Puddle and Pine Lands had similar initial concentrations of ammonia and thus had similar percentages of growth. However, the variations of growth can be accounted for the fact that very small masses of duckweed were used. Some even had only 0.3 grams of duckweed, meaning variations in mass greatly affected the percentage of growth. The conclusion that duckweed grows far better in ammonia-rich media demonstrates that duckweed can be a highly effective wastewater remediator as it grows better and absorbs more nutrients, as is visible in this chart.
So our second experiment, the nutrient content examination, further tested, along with what Nikita was saying, about using duckweed as a wastewater remediator and ability to curb eutrophication, hopefully. So what we did was we originally took water sources, or samples from a water source, the Princeton Meadows Municipal Water Treatment Center, and we tested them for their original levels of ammonia and nitrogen. So using that as our control, we then inoculated seven different species of duckweed into other water samples from Princeton Meadows and tracked the actual nitrogen and ammonia levels over the course of 2 weeks in that media. So then we compared that to the control so we could see if there were some sort of changes. So for our original sample from the ammonia, there were 0.20 milligrams of ammonia per liter of wastewater, which is actually very high, wastewater treatment center. So then we compared that to our actual results into, as you can see in this chart, each of our 7 different samples, which had the 7 different species of duckweed. And actually, in every single species, there was a 50% decrease in ammonia and nitrogen levels over the course of 2 weeks. I'd like to specifically point out sample number 3, right there, because it actually saw a 50% decrease in ammonia and nitrogen levels every single week. So by the end of 2 weeks, the ammonia levels dropped from 0.20 milligrams per liter to 0.05 milligrams of ammonia per liter, which thus illustrates duckweed's ability to be used as a wastewater remediator.
The final procedure involved the starch assay. So I'll begin by just talking about the procedure of ethanol production first. So yeast, through fermentation, takes broken down simple sugars, generally glucose, or in our case dextrose, consumes them, then emits 2 waste products, one being carbon dioxide and the other being ethanol. So in our starch assay experiment, we first took grinded duckweed, and then we added a buffer to stabilize the pH level. After that, we autoclaved it to maintain a stable environment. We then added an enzyme that catalyzed a hydrolysis, which essentially means that starch was converted into dextrose, our final product, that was then inoculated and centrifuged before being harvested and analyzed. So, in our analysis, we found that approximately 9.68% of duckweed is composed of dextrose. In comparison, only 4 to 6% of corn is dextrose, proving that duckweed, even in such a small biomass, is far more efficient in producing ethanol, making it a much more viable source.
So it's really a shame in the United States currently that our major source of ethanol and biofuel is really corn or maize. Because with the skyrocketing crop prices of corn, meshed with the other economic and weather-related difficulties with corn production and crop production, there has been a huge discouragement in the cultivation and growth of biofuels. So researchers should really now take a look at duckweed, because as we have shown, duckweed can be used as a source of ethanol, highly efficient, and can actually supplement corn-based ethanol. Additionally, because it can thrive in such toxic chemicals, it can also be used as a wastewater remediator on a larger scale.
So again, to exemplify Anne-Marie's points, duckweed is a presently optimal solution to solve both the crises of wastewater and energy needs. However, future research can still be conducted to see how the plant fares in varying conditions such as different surface areas or climates. However, previous research has already shown that duckweed is extremely resistant to high salinity levels as well as areas of low sunlight, making it very easy to globally harvest. Other research can also be done to see how duckweed can remediate different affected water sources with different phosphates or toxins. Again, although the economy is currently recovering again, a near future scientific research endeavor may not be possible. However, it would be definitely optimal to have a new market with duckweed-based ethanol, shown with its optimal nature. In our research, STAR is definitely being substantiated as a source of ethanol production.
So right now, we'd just like to thank these following people for, if they were not helping us along through the month of July, we would not be doing this research nor presenting in front of you today. So specifically, we'd like to thank Dr. Eric Lamb, Dr. Nikolai Borshchuk, Jorge Montalvo, Philomena Shu, Dina Eileen Rosen, Jean Patrick Antoine, the Governor's School Board of Overseers, Tara Nealon, South Jersey Industries, PSE&G, Rutgers University, the State of New Jersey, Morgan Stanley, Lockheed Martin, and the entire GSET staff and community for their continued support.